Rick.
Hello and welcome to those of you who are still already with us. We are waiting our second speaker and some other people joining quietly the webinar, so be patient. I apologize if my video camera doesn't work. I'm sorry, we have some problems. It won't be long. Our second speaker is joining shortly. I must apologize with all of you with this delay, but we experienced a national network problem just half an hour ago. So we needed to set everything in place rather quickly. So be patient, a few minutes. Hi, Luca. Here we are.
Hello.
Hey. Hello. Hello, everyone. I think that we can start if we have the go ahead for the YouTube channel.
Sorry, I had some problems with the connection.
Okay, no problem. Now we are all here. So thank you for all of you for joining this webinar. I'm Giovanna Cicognani from DLL, Alessandro Tengattini and Lucas Helfer, our speaker today. As most of you certainly know, this webinar is organized in the framework of Science in School, which is a journal for teachers and students funded by the Aeroforum organization, which are all the European research centers in Europe. And this included the ILL, which is the host today. The mission of Aeroforum partners is to promote by other missions several outreach activities, such as, for example, the production of the Science in School journal, and also a series of webinars like this one, which are meant to give teachers and students the opportunity to hear directly from some of our scientists what they are doing, the cutting edge of the European research. So as I said, today's webinar is hosted by the LL, which is a research institute producing high-intense neutron beams used to explore matter, to understand its properties. And as an example today, our speakers will show you our tomography and imaging facilities allows scientists all over the world to obtain 3D images of their samples. So I'm happy to introduce you Lucas Helfen and Alessandro Tengatini, who are scientists working at the next instrument, the tomography station at the LL. Alessandro is also an associate professor at the University Grenoble-Alpes, where he focuses on couple process in porous media. And Lucas, before joining the LL in 2019, was a scientist at Beamline at the SREF, which is an European research center, a synchrotron here, close to the LL. And his research is directed towards energy materials. So I let them speak now and enjoy the talks.
Wonderful. So you should now see my screen, hopefully. Essentially, what we'd like to talk to you about today is tomography. So the 30-second explanation of what tomography is essentially an extension of 2D radiography. We're all familiar with radiography. If you go to the hospital, hopefully not, but you probably are aware of it. You can take a radiography of your hand, and essentially what you'll mostly see is the bones because they are a bit more dense. So the tomography extension is just, instead of taking just one projection along one plane, you can acquire different projections along different planes. And once you have all this angular information about all these different projections, you can back project it, as we'll see much more in detail later, and get a 3D image. Now, this 3D image is not just the outer surface of our object, it's not just the bones we see, but it is in general a bit of everything that is inside, with different kinds of contrast, as we will study in detail. But ultimately, what it is a 3D matrix, a 3D array of attenuation coefficient. Now, this might sound a bit confusing, but we'll get much more in detail, and hopefully this, what I just said, is going to become much more clear. So let's start from this simple 2D example. You take you broke a bone, let's say, you implant a titanium implant there, and this is what a radiography will look like. But if we try to zoom in a little bit, you can start seeing pixelated details. And if we zoom in even further, what it is essentially just a 2D grid of values. Now, what are these values? Well, values for a computer are just numbers. It doesn't understand anything else, really. It can only understand numbers. And it is us typically telling to the screen or to the printer, well, this value, which is, I don't know, 220, is assigned to this specific, it looks this gray. And this other value, which is 110, is a lot darker. And when we adjust the brightness and contrast in a phone or in a computer screen, all we're saying is that, well, I'd like your brightest bright to be this number and your darkest dark to be this other number. Now, this is a 2D radiography, essentially. It's just a 2D array. It's a 2D composition, a puzzle, in a sense, of different attenuation values, and we'll see exactly what they mean. But what I'd like to specify is that the extension to 3D is actually, in a sense, quite banal. It's instead of being just a 2D array, a 2D puzzle, it's a 3D puzzle. It's a little cubes, right? So the same values applied for a certain area of space, which is not anymore just a square, as in a pixel, but it's going to be a 3D, so a voxel. Instead of being picture per element, pixel, it's going to be voxel, so volume per element, voxel. What do those values mean? Now, we've seen 110. What does that represent? Well, it depends on what you are illuminating your object with. So, for example, in the case of X-rays, what X-rays interact with is the outer electron shell of the atom. So, as you probably know, an atom is made of a nucleus which contains protons and neutrons and a number of electrons just spinning around it. Because the X-rays interact with this outer electron shell, so these electrons spinning around the atom, the more electrons we have, the more this object will look opaque. So, for example, if you go from very light elements such as hydrogen, air, or these kind of things, you will have that the objects are mostly transparent to X-rays. And as you start going towards heavier and heavier elements, so metals, uranium, these kind of things, they become more and more opaque because you keep adding these electrons around the atom. Now, in the case of neutrons, this is not as intuitive because the neutrons will interact with the nucleus of the atom. So you have very light elements such as hydrogen and boron, which are very transparent to X-rays, and they're very opaque to neutrons. And vice versa, you have very heavy elements such as lead, which are almost transparent to neutrons. So it's a bit less intuitive, and we'll get more in detail to why and when we can exploit this interesting difference. If you look at the periodic table, it repeats essentially what we just said. If you go from very light elements, or just, for example, hydrogen, which only has a proton and an electron spinning around it, it's very light, it's very transparent. And as I go down the periodic table, it keeps on increasing the opacity. And conversely, when I look into the neutron part, I have very light elements that are very opaque and very heavy elements that are quite transparent. It is an important complementarity of these two techniques. What does it mean in reality? Well, let's take a picture of the camera, for example. And in the X-rays, you can see here, all the plastic parts are quite transparent. All the metal bits, though, they become very, very, very opaque. Conversely, when we take the same image in neutrons, all the metals, they become almost transparent all of a sudden. But the plastic bits, for example, the plastic of the flash, the plastic of the cover, or the lithium contained in the battery that powers the camera, they are very, very opaque. Let's take a completely different material that you are probably sitting on in a way or another. It's concrete. It's the most used fluid in the world after water. It's everywhere, and we make a huge amount, use a huge amount per year. What is it made of? It's made of little aggregates, so just essentially just large grains of sand, a cement paste that binds them together, and there's a lot of little holes, which we call pores. Now, when we take a tomography of this object, we can take, for example, a horizontal slice just to see what it looks like of this tomography. And we can see that it's very easy to see the pores because they are transparent. It's just air. So we said air is transparent to X-rays. But conversely, the grains in the cement are very, very similar. They have the same opacity. But when we do the same thing with neutrons, all of a sudden, the grains, because they are made of quartz and oxygen, they are very transparent. So paradoxically, it's very easy to distinguish the cement from the grains, but all of a sudden it's the pores and the grain that are too similar to be able to easily distinguish. So you can start seeing that these two techniques are incredibly complementary. So it is only sometimes using both techniques that I can understand completely what an object is made of, looking inside the object and recognizing the different components. And understanding what an object is made of is very often the essential step to try and characterize it and make it better and improve it. So how does a tomography actually work? Let's take a very simple material, just a sand. You can go to a garden, you can go to the beach and get a little bit of sand. And all of a sudden you put it in an X-ray tomograph, for example, in which you'll have a little source here on the left, and you'll have a detector there on the right. And this will shoot a number of rays, which are just my individual photons, and will shoot a lot of them, and will create a nice little cone, for example, in a typical hospital scanner. Now we put our object inside our scanner, we have characterized what the beam looks like, and all of a sudden we impact this object with a number of photons, essentially. X-rays are just photons. These photons will penetrate a little bit through our object. Some of them will manage to go through, and some will just get absorbed or bounced off. So eventually, what I have on my detector, what I can measure on the other side is a variation in the opacity. just as like in the hospital, if you take a horizontal slice of this object, you say that, well, it's very transparent, and then there's a bone, so not very transparent, and then it's a bit more air, and then there's a bone, another bone, and there's a very transparent. So this alternation is essentially a line of pixel in our acquisition. But this is a radiography, essentially. You're just measuring how many of your photons survive. You have a certain amount getting in, some of them will not survive, and you're trying to see how many get to the other end of your sample. Now, all you need to do is to repeat the operation in a number of different angles. And you can see here that depending on the angle, the radiography will look different, of course. And once you've acquired enough angles, then you'll have a large field of information. So this is enough. You acquire a number of projections at different angles. How does it work, though, to reconstruct a 3D object? How do we rebuild our 3D image? Well, let's work backwards. Instead of trying to, now from this information we accumulated, we know that in this area, for example, in this little bump here, it means that there's going to be some material here on the back somewhere. We don't know where it is at the moment. So let's just assume that it's homogeneously distributed along that in between the detector and back in the source. Same here, there's another little bump. So we know that along this bump somewhere, there's going to be some information, right? So we are what we call back projecting. We have this information projected on a plane, and we are saying, I don't know where it is, but it's going to be somewhere there. Now we repeat this operation. We acquire not just this angle, we acquire a number of angles, right? So we can accumulate all this information from all the angles we have accumulated. And if you have a good eye, you can start seeing that here in the middle, we already start building a little cylinder, and our sample was somewhat cylindrical. And you can already see that there's areas where definitely have a little bit more attenuation. It's a little bit more opaque than in its neighbors. All we need to do now is to have enough information. And for example, with 17 projection, this is what it looks like. You can kind of tell that there's a cylinder somewhere, but it's very, very noisy. It's very messy. But as I start adding more and more projections, adding more and more information from more angular position, all of a sudden I can start seeing a bit more until when you have, let's say, about 1,000 projection. Well, here's an object. You have a 3D information. I'm just representing a horizontal slice of it, but it's a full 3D volume. And you can see that this little object is made of tiny little grains that just a sample of sand, in this case, contained within a cylinder. Now, you can already see by looking at these images that an important aspect when looking at an image is its quality. And we're not going to get into the details of image quality, but there's a number of things that can characterize. One is the signal to noise ratio. So essentially how much information I have. A simple way of looking at it is when you take a picture in the dark at night, you'll see it's very grainy. There's a lot of noise, and that's because the ratio between the background noise, the natural noise of the graininess of your camera itself, is very close to the signal itself. Whereas if I take a picture in the middle of the day, even if the exposure is very short, my signal will be nice and crisp. But there's also other factors such as the blurring. If you have taken a camera and you have it a little bit out of focus, or there's a smudge on the lens, it's going to look blurry. So there's a number of things that needs to be optimized when acquiring an image to get a nice, crisp object. Another important thing is the trade-off between the resolution and the size of your object. So if, for example, thinking of a lab scanner, of a hospital scanner, if I take my object very, very close to the source in this case, I will have a very high resolution. I'll be able to see tiny details of my object, but I will not be able to see many of these grains, many of these objects. Why? Because, well, my detector will only have a limited number of pixels, will only have a limited number of chunks of information. I can do the opposite. I can have a much coarser idea of a much larger object, but of course my resolution is gonna suffer. So there's always a trade-off between how accurately you see something and how much of it you can see. And which one of the two is interesting? It really depends on your problem, because if you look at the sand at the level of a whole coast, well, you don't really care about the individual grains. But if you start trying to understand how the different grains interact together, well, you better have an image in which you can see the grains. And if you want to pass to the step after, it's trying to say, okay, maybe in some conditions these grains can break. For example, if you build a house on top of them, they would start breaking. Well, then you need a very high resolution. You need to focus on a much tinier area. So depending on your problem, you can study the same thing at different resolution, but resolution is not the only parameter. As you've seen, you might want to have more of them at lower resolution or fewer of them at much more high quality. Okay, we've mostly seen examples now about X-rays, but we were talking mostly about neutrons today. We're a neutron center after all. So what are the advantages of neutrons? Well, I imagine that you know already a little bit the X-rays. You've seen some images of bones or of the inside of an object, but why use neutrons? Well, the truth is that neutrons generally have a much higher penetration power. So, for example, if I have an object within lead, I can irradiate it for essentially hours, and I can barely go through. But if I repeat the same operation with neutrons, after a few seconds, I'm fine. I can just penetrate. Also, there are some elements such as hydrogen and lithium, which are very transparent to X-rays, which are very, very opaque to neutrons. And this brings to some beautifully paradoxical images, like if you put a flower behind a layer of granite, which is very dense, which is very hard to penetrate with X-rays, essentially you barely see it with the granite with neutrons, and you'll see very nicely the flower. Or if you take a flower and you put it in a container made of lead, you can see the flower and barely can see the lead, which is in some cases very important to have this alternative penetration. There's very low radiation damage. As you probably know, you don't want to spend too much time behind an X-ray machine because it can be quite aggressive. It can damage a little bit your the DNA and the tissue in your body. Neutrons are a little bit more gentle in this respect. I still wouldn't recommend a person to do it, but if you're trying to study, for example, plants, X-rays can really affect their behavior, whereas neutrons will tend to be a lot more gentle with them. And also we are isotope sensitive. This is a slightly more complicated thing, but we'll see it more in detail. But essentially there are elements which have more or less neutrons in their nucleus, and those are called isotopes. And because we interact with the nucleus and not with the electron shell, elements which are very, very similar from a chemical and a physical point of view will have possibly very, very different attenuations. And that's the case, for example, of normal water and heavy water. But we'll see that more in detail later. And finally, neutrons can directly see a magnetic field. So there are ways in which you can directly see essentially how a magnet warps the magnetic field around it. And it's very important in some cases to really understand them. We can get 3D images of this. We can really get not just the amount of the magnetic field, but its direction and its orientation in space in 3D. So a full 3D image of vectors. So let's take a very simple example to try and understand the difference between X-rays and neutrons. The simplest example in a porous media you can think of is I'll take a rock and I put it in contact with some water. Now, as you probably know from going to the beach, as soon as you put something porous or a sponge, for example, next to water, by capillarity will tend to suck up the water. And if I try to take an image of this with X-rays, what I see is not a lot. I can barely see the front, whereas neutron is nice and visible. So let's see what it looks like, for example, the inside of a coffee machine. If you've never seen it, you can see this is a radiography of a coffee machine working, and you can see here immediately the plastic of the handle and of the nib at the top are very, very visible because they're full of hydrogen. Here the water, you can see it as it starts boiling, and it will build up pressure here in the corner, and this pressure will push down the water, and there's a nozzle going down. And this nozzle will be the only way for this water to escape. So the water will boil, will go through this nozzle. And here the interface will find the coffee itself, and it will go through it with a very high pressure and very high temperature, which is what manages to take the caffeine out and get the nice little coffee out which you want. And you can see here, as it goes to the top, this boiling water gets into a colder environment, so it starts redepositing. And you see it's not a very stable process, and which is why it makes this very peculiar noise in the morning if you have one of these, let's call them Italian coffee machines. And as the coffee goes up, it will redeposit and will depose. And now it's not just water, now it's water with a lot of caffeine and a very nice taste, hopefully. But this is a very simple example, but let's go back to the example of our rock. What can we measure? What are we interested in? Well, we can try to look for where the front is, right? We can try to push this water through and try to see where my front is, because that will tell me something about the rock itself, which apparently quite a few videos are not going to be working. But essentially, my front is going to be, we're going to be able to track where the position of the front is and how much in depth this can go. This problem becomes even more interesting when we have cracks, because if it's a very homogeneous front, a flat front, I don't really need imaging, right? I can just push some water and try to wait at the top when the water comes in. But if the rock is not homogeneous, if there is, for example, a localized deformation, as it often happens in real materials, then all of a sudden I can see how the different the fractures and fissures will affect the evolution of my front. And essentially, what we get here is a 3D speed field and a deviatoric strain field. So by comparing images at subsequent steps, what I can get is how the object has deformed. So if I load it, it might deform homogeneously or very heterogeneously. So I can compare where the water goes to how deformed my material is. And that's something you can only see when we have any information about the inside of the material. But let's now go to slightly more complex geomaterial. I can try to push some water into something which is real, a real rock taken from a mine 3 or 4 kilometers down the crust of the earth. And why is this interesting? Well, this is interesting because oftentimes those rocks contain something that we want. It might be fresh water, and we might be pushing it with salt water because we want to extract it to give water to people. Or it might contain hydrocarbons, which we use to power our vehicles and power our entire society. And you might want to see how effectively you can push this water, you can push this oil through a rock. And the truth is that the images we saw before were nice homogeneous rock, but the truth is a lot more complicated. And you see here, as I start injecting, the water will mostly go through the fissures. It will accumulate in certain regions which are really thirsty of water. They really want to keep the water, typically when there's clay and this kind of material. But the truth is that this was a dry rock, so it was a complete dry rock in which I've pushed a fluid. In truth, in reality, oftentimes these rocks already have a little bit of fluid inside. For example, the fluid I want to extract. What then? What can we do? Well, we mentioned before that neutrons are isotope sensitive, which means what? Which means that if I take a normal hydrogen and a so-called deuterium, which is the same thing, it just has a proton and an electron going inside, is just that in deuterium, there's also a neutron going around it. And because the elements interact mostly at the level of the outer electron shell, these two atoms will behave very, very similarly. And as I can make water from hydrogen. So I take two atoms of hydrogen and one atom of oxygen and put them together and make my everyday water. If I take two atoms of deuterium and I put a normal oxygen inside, I will make so-called heavy water, which is like normal water, just a little bit denser. But the interesting thing from a neutron point of view is that they will have very, very different opacity. They will be very different for the neutron. So that the first flash I've done here actually was done with the heavy water. But then now the fluid is saturated and I can push it again. And despite the fact that these two fluids are the same, they will look very different. So if I take the difference of the images, I can see here that the behavior of my fluid is very different. It's not going to be accumulating anymore here. It will just pass through this fracture system. So the behavior, it can be very, very profoundly different between the two. What happens in 3D then? Well, in 3D, I have the same information, not just anymore the projection we were seeing, but I have a full 3D map of what's happening inside. Okay, let's take a different example. Same idea, but for example, a plant. Why might I want to study a plant? Well, the truth is that the plant affects very severely the soil around it because it tries to suck out water locally around the roots. I have significant variation of the permeability, so how well the soil will allow the water to go through. And this process is very, very fast because if you ever try to give water to a plant, the water gets sucked up very, very quickly. So we need to acquire our tomographies very, very fast, which is, oh boy, which is what we would have seen here. If you give me a second, I will try to share the full screen so I can show you the video. I will share directly desktop. My apologies for this little hiccup. Lots of videos I would have liked to show you, but and here we apologies, just a second for sure. And here we go. This is what the plant looks like. These are very fast tomographies. Each one of these images is just 1 second in time. And you can see here the fronts progressing. I would normally be able to see. Here you go. This is the one. And here you go. You can finally see the front of the water being pushed, being sucked up by the soil upwards and moving very fast. So very fast tomography, just 1 second per tomography. Fantastic. Let's go back to our rest. So I hope I convinced you somewhat that neutrons have a very different contrast from X-rays, and that neutrons can see light elements such as lithium and hydrogen. They are isotope sensitive, and we can go through a lot of metal. So these are some of the unique capacities of neutrons. But sometimes the question is not whether we should use neutrons or X-rays, but whether we cannot use neutrons and X-rays. So because the two techniques are so very complementary, sometimes it's only when we join these two information that I can get a complete picture of what's happening inside. This is a bit the logic behind the instrument which we have built here, which I'll show you a little video of. With a bit of luck. Which is this one. So this is what an instrument like that looks like. So we can see here there's a lot of lead outside, and there's a lot of concrete in the back. There's of course a steel structure that holds it all together. And you can see here there's the neutrons, this blue line which comes down from the reactor. There's a reactor that will produce all these neutrons, and there's a number of instrumentation we'll try to very quickly go through. But there's a number of things we can do to the neutrons to change a bit their nature. But let's try to see if we were a neutron, what we would see on an everyday instrument. So we would go down. There's of course all sorts of safety shutters that will block the neutrons when we don't want to see them. We can see these little pinholes here. Our objective allows us to make the neutron nice and parallel. And Lucas will get more in detail on when and how much we can collimate. We can select just a small amount of neutrons to make sure we have the most parallel one. And we have all sorts of things such as interferometry here to change the type of contrast. And we can see here that this, if we were the neutrons, we would just impact against our detector. But you can see here there's an X-ray source and an X-ray detector which is perpendicular to it. And these two can be acquired fully simultaneously and can provide very different information. And there's of course different size of detectors. We can see here a big detector and a small detector. As we saw, as we mentioned before, we can have higher resolution or lower resolution for smaller or bigger objects. But let's have a look at why, in what cases we can use neutrons and X-rays together. Well, a very simple example is the one of, once again, rocks. We've seen before that if you put water in some rocks, their behavior will be essentially, the rock will not move, they will not be affected. But there are some rocks in which as soon as you put a little bit of water, the behavior will be significantly different. I'll try to show you a video. And here it goes. And you can see as soon as we put in contact our rock with water, it will break essentially. And this is very important because maybe you have put something delicate in that rock. Maybe you want to store something like nuclear waste and like that, and such things in this rock. So you need to be to truly understand accurately how the rock will behave when we put it in contact with water. And we see here at the top with X-rays, you can see very clearly the opening of the fractures. And that's something very important, but we cannot see what causes this fracture, which is the water. And conversely, when we look at the neutron image, which is here at the bottom, we can see very clearly where the water is. That's the stuff in white, but you cannot see the fractures so well because the water is filling them up. So if you want to understand really the combination of the behavior of the water and of the rock, then you need to acquire the two at the same time and join this information. And there's all sorts of questions to be asked, like how, depending on how this rock was deposited, this fluid will go predominantly along the fractures or orthogonally to this, perpendicular to these fractures, which is what we see here. But let's now go back to the example of concrete. Once again, it's an object which is used in everyday life. We are all, in a way or another, sitting on it today. And once again, neither technique by itself can tell us everything about it, because as we saw before, in X-rays, it's very easy to find the pores these little holes in the concrete, which are perfectly natural and perfectly fine. But it's very hard to distinguish the other two components, which are the cement paste and the aggregates, so the large chunks of rock that are inside the concrete. Conversely, in neutrons, you can see very well this cement paste because it's full of hydrogen, but both the pores and the aggregates are somewhat transparent. So it's only when we combine together these two information that we can do what we call a segmentation, which is being able to accurately say, well, this bit of my concrete is an aggregate, this bit is a cement paste. And then I can, for example, thermally, that's not my day, we can combine these two information, and you can see that with the X-rays, it's very easy to find where the fissures open, and with the neutrons, we can see the drying of the concrete. For example, I might be wanting to heat the concrete to see how it behaves in fire conditions. So I want to see how fast it will dry, because this is what's going to determine whether a building, when it catches fire, will collapse or will be perfectly safe. And we always want, of course, to try and improve the way in which we make our building safer for everybody inside. And once again, the X-rays will provide very useful information about the evolution of the microstructure, in this case, the opening of these fissures, whereas the neutron will tell us where the water goes. Fantastic. And I can study, for example, how these different fissures, this little crack that opens in the concrete as we heat it up, will make this process faster or slower. And then I can compare all this information to my nice little models. That's it. So these were hopefully a couple of examples, but my colleague Lucas will go through other examples from our everyday life, for example, batteries. But I hope I convinced you that there are these two techniques combined are very powerful together, so neutrons and X-rays. And that a few details that we can go to relatively high resolution. We can see objects which are 4 microns, so 4,000th of a millimeter, so tiny, tiny objects. And there's a lot of fun to be had ahead. There's a few resources about neutrons and imaging in general, and of course there's a website we have about the instrument. You can go there, and there's a little bit more information that we had the time to go through. So now I'll pass the word to Lucas.
Okay, thank you, Alessandro, for this very nice opening of the topic. I hope you hear me all. Please complain if you don't. So I will share my screen. And so just going to presentation mode. No, doesn't it work? Right. Yes, after this very nice introduction on what you can do with the neutron imaging, I would like to go a step back and ask ourselves, why do we do that? So what can be the motivation for structure characterization by neutron imaging? There is of course, to get information about the structure itself. So for instance, get statistical parameters that we want to get out of the structure. We would like to see if there are imperfections, perhaps due to production flaws, and that we need to avoid during production of structures and materials. And then, of course, we would like to see the relationship between structure and properties, as we've seen in Alessandro's talk, that the permeability of a stone or of some cement, how is that affected by the microstructure? So microstructure to the macroscopic properties. If we manage to do that correctly, then we can model that for simulations, and we can then afterwards close the loop somehow and perform virtual materials design by simulations on the simulated materials, and then deciding which has the best macroscopic properties, and somehow try to optimize these materials by computing. Of course, there's the other aspect of looking at the temporal evolution of structure, what has already impressively shown by Alessandro. Sorry. For instance, if you put a waterfront under the concrete or rocks, or if you put a particle stream through porous structure here, for instance, if you thermally or mechanically treat the objects or structures, Or if you want to see working devices operando, that means that you do an experiment during the device operation. For instance, also during a battery that discharges or somehow a current that goes through the device and degrades it, for instance, in electromigration. And of course, the aim is to provide a non-destructive characterization in order to be able to follow the evolution of processes. So, and for that, a neutron or X-ray imaging, as Alessandro has already shown, are very nice methods, because you get projection images in the radiography. So you see what happens inside the structure, and if you afterwards combine it with cross-sectional tomographic imaging, taking different viewing directions of the sample, as Sandro has already shown, then we can get to cross-sectional imaging without destruction of the sample, and to 3D imaging, so that you have any possible direction of cut through the sample that you can look at and even see the structures in 3D. So Alessandro has already shown the chemical table of elements with the gray values showing the relative absorption. So here I show the relative absorption, or the mass attenuation coefficient comparison between thermal neutrons and X-rays, here 100 keV X-rays. The X-rays, as Alessandro has already said, they are interacting with the electron shell of the atom, while the neutrons interact with the nucleus. And what we see is we have this Z dependence of it's a continuous dependence in the X-rays, but the neutron cross sections, they are very much scattered. So you can see very well distinguished in many instances neighboring atoms with neighboring atomic number Z. So, and also, as already pointed out by Alessandro, you can very well distinguish the hydrogen, for instance, by its isotope deuterium, which is just a proton with a neutron in the nucleus. And so you get isotope sensitivity. And so this you can, for instance, see also with lithium. Here you have natural lithium, sorry. And the 6 isotope of lithium has a much higher scattering cross-section or cross-section than the natural lithium. And here Alessandro has mostly shown things on hydrogen and water. I will go more here into lithium and the distinction between lithium from the surroundings, where we see that with X-rays, if you have carbon, for instance, or hydrogen or nitrogen, oxygen inside a component, then, or in a material, then you can very badly distinguish it from one another. And with the neutrons, we are well sensitive to the lithium due to its high attenuation coefficient. So I show that here with the lithium-ion battery principle. So that is a principle that was invented, or let's say developed, since the 1970s, 1980s. And in 2019, these 3 gentlemen got the Nobel Prize for it in chemistry. So the principle of the battery is that of an ion transfer cell. That means you have here during the charging, you store the lithium in petroleum or graphite in the interlayers here, and upon discharging, it releases an electron, and the ion goes through a separator, a barrier, via an electrolyte, and goes to the cathode. Sorry, that was a bit too fast. And the cathode, they're the same things, same principle applies again, that the ion gets intercalated into this NMC, for instance, materials, or cobalt oxide cathode material. So for the intercalation, you generally create stresses and volume expansion, and that makes you on different charging and discharging cycles, the material works and expands and contracts. I will show you what you can do now here with a working cell. So operando discharge of a primary cell here of a thionyl chloride cell. It has a liquid electrolytes, which is in a porous carbon structure for the electrical conductivity. And we have a pure metal anode, lithium metal anode, which is connected to the outer casing here and provides negative electrode. And the carbon structure is connected to the positive electrode here. So what happens when we discharge the battery? We see that the electrolyte, the liquid electrolyte, and also the cathode itself gets consumed. So there's SO2 gas and sulfur being produced, and we see that here then the lithium goes away from the lithium anode metal to these compounds that they are here forming in the former cathode material. If we go through here, so the first stage and the last stage are the reconstructed slides that I've shown in the movie. So it's not a superimposed image; it's a reconstructed slice. Temporal resolution is something like half an hour here in this case for rather slow discharging of these cells. And so what you can see is how the lithium metal here, that is a spirally wound bobbin type structure, gets discharged and gets then to the thionyl chloride cathode material. So we see that here in 3D that the anode dissolves, and in the end you get even bits that are not connected anymore to the main anode material, so to the outer pole. And we can then see how is the lithium metal consumption and how that relates to the capacity that we are drawing out of the battery.
Recently emerging multi-beam setups for tomography require no sample.
You can do that for different currents. For instance, the different discharge currents here are 300 milliampere and 100 milliampere discharge currents, and you get then to something like a capacity of something like 1.4, 1.2 milliampere hours that you can draw out of the battery. The same here is now for the different materials. The manganese oxide primary cell again, so you cannot charge it; you can only discharge it. Here now with a combination of X-ray and Neutron, because here in the X-rays we see very well the nickel mesh that supports the anode, the cathode material, and again here the lithium metal that gets dissolved, and the electrolyte gets consumed here, and we see then the swelling of the cathode material, and that gives then rise to cracking in the electrodes. So these here were experiments that have been done at HCB. I just show you, but it gives you a rough idea what you can do on the scale of some 20 minutes, 1 hour scan time, half an hour, let's say. If you want to go to higher spatial resolution, then you need to. So if you want to see, for instance, what happens inside exactly the anode or cathode materials for rechargeable batteries, then you need to really to make a good compromise between spatial resolution and temporal resolution. And what we are using at NEXT, and what many other neutron imaging instruments do on the world, they use indirect detection schemes. That means that you use a scintillator material in order to convert the neutrons to visible light. The visible light gets collected by optics via a mirror in order to get the sensor to the imaging sensor out of the transmitted beam direction. And you have lenses in order to focus the image onto the camera sensor. You can have two different schemes. So one here with a non-infinity corrected setup that you focus on the screen via one lens or one lens system. Sorry. Or you can use the infinity corrected approach where you have where you have a collection lens after the scintillator. And this collection lens gives you a much higher numerical aperture and light collection efficiency than the other approach. So it's more adapted to high resolution imaging. Also, the scintillator must be finer because you, with this high numerical apertures, you are getting less depth of field. That means the scintillators needs to get thinner. And there are different approaches. So you could, for instance, say, okay, I deposit a powder screen on a substrate, and then if you look then at the microstructure with a microscope, you see that there's inhomogeneous illumination of the camera signal that it can be sometimes a bit bothersome. Or you could have, for instance, a doped layer of a crystal, of a single crystal material, for instance, produced by liquid phase epitaxy on a substrate. And this is the case here of the GGG crystal. You observe then the scintillation processes via the substrate here. So you can have then secondary effects also. Here you can have, for instance, multiple reflections between the inside and outside of the between the two insides of the substrates. Here on the powder screens, you have scattering taking place in between the powder particles. And so normally, if you want to go to high resolution, like 5 microns, you don't want to have powder particles larger than that. And the general thickness of the screen also in the order of some microns then. Spatial resolution does not only depend on the detector system, but also on the collimation of the beam. So if you're looking at the neutron guide or at the cold source, they are very low brilliance sources. So that means that the source, the emitting source, is quite large. You can see the easiest way of dealing with that you introduce a pinhole. So you have here a neutron guide getting the neutrons from the cold source to the imaging experiment. You create a total external reflection here in order to keep it in the guide, and then you have the diameter of the pinhole that defines you the collimation ratio together with the distance where you put the sample in the end, the distance L here. And so the problem is, of course, when you put the detector at a finite distance, you will get some blurring, which depends on the collimation ratio that I've just said. So the relationship, because of the similarity of these triangles, is that L divided by D, the diameter of the source of the pinhole, is the same as the blur circle, as the detector distance divided by the blur circles. And that means, in practice, in order not to sacrifice too much neutron flux, because if you close the D, the large D, capital D, then it goes neutron flux goes down, and so you in practice you're staying in something like 300, 600, whatever, in order to get enough collimations. Of course, it depends on afterwards how close we can go with the detection plane towards the sample. And if you do tomography, you turn the sample, you have the ideal case of a cylinder. Then you can really approach the detector, the detection plane, as much as possible. If you don't have a sample environment, otherwise you need to have the distance of the sample environment that comes into account. And let's say if you have 1,000 pixels that you want to reconstruct, then you would say, okay, I can accept something like 1 pixel of blur radius, in order not to be less resolving than the detector. And so what we see here, we get already with the 300, we get something like 3 milliwatt. That means that that in practice it doesn't hurt too much if we get blur circles in the order of 2 to 3 pixels. Okay, I show you now some examples also here again coin cell batteries that you can have a look at, and look at the microstructure of the cathode and anode materials. So at next we can go with very high spatial resolution. So here, for instance, the pixel size in the order of 4.2 microns with long scans. That's the inconvenience. So it is not so much adapted to operando and imaging during discharge of batteries on charging cycles. But you can then do studies on of the aging of the capacity fade during aging of batteries. So if you charge them 100 times or 300 times, what happens then with the cathode and anode materials? For that, as already shown by Alessandro, we can use both X-rays and neutrons in order to very well characterize the different components of the battery. So here the X-ray image doesn't need to be done with the same pixel size or sampling, spatial sampling. So here we just increase the pixel size and turn the image right in order to make it approximately match here. And then we can create a composite image where you show here the highly absorbing X-ray in red and the highly absorbing neutron in blue. For that, you can use an extension of digital volume correlation, that is a multi-model registration. So you somehow define a cost function, and then iteratively you align the images by getting to an extremum of this cost function, which depends on the joint or bimodal histogram of your sample. I don't go much into detail here, but what you get out then is then the aligned image. So you see then very nicely highly X-ray absorbing structures here. X-ray absorbing structures in red. That's the copper current collector here, for instance, this small stripe here. And the casing, of course, which is made of stainless steel. And you get highly neutron absorbing structures here, for instance, the lithium containing cathode material here that is in a discharge state. And what you can then do in order to get to profiles across the battery and to show the different charging states is that you can virtually unroll the battery. You can do a polar transform and then put the different layers in a polar diagram here, and you can then see that the different layers and the local fluctuations of the layers. And you can do that, of course, for the discharge state here, for instance, after 700 discharging charging cycles. And you can do the same thing for the charge one, which is here in the bottom, with the contrast increased. And if you zoom in some interesting regions, we see that there's a lot of variation. And this variation is due to the discharging cycle. So the material ages, and the lithium is trapped here in the discharge state in the anode material, for instance. And we get also voids or weakly lithiated regions here, both in the anode and in the cathode material in the charge state. So that we can then look the different cases that we can see. We have here, for instance, voids that appear in the anode material. We have plating of the separator materials, for instance, here between the anode and the cathode that can occur. Or we can see asymmetries between the two, between the inside and the outside anode, because it's a sandwich structure, which is where you have here an anode material here on both sides of the copper current collector. And then, of course, you can try to correlate that with simulations and try to figure out what are the different, what is the theoretical attenuation coefficient that you would expect, and try to model that and with the measured plots across these profiles here. Okay, that was my just a concluding remark here on what you can do with imaging. I just showed you one small part here of lithium transport in batteries that is due to the high sensitivity of the neutrons towards lithium isotopes. You can have a look at bone structures. You can look at devices from particle filters, where you can look at soot inside the filter structures. Sorry. And as Alessandro has already shown, the water uptake in plants, hydrogen, for instance, in metals, that all basis is based on the attenuation of neutrons. That means of the reduced transmission in the forward direction of the field, but you get also phase effects due to, for instance, magnetic contributions. So you can have a look at magnetic fields, as Alessandro pointed already out in the beginning. We can look at magnetic domains because the magnetic domains can depolarize the polarized neutron beams, for instance, and we can then see or have a look at internal defects in for instance, generator steel materials. And by diffraction, combining it with diffraction in the monochromatic beam, we can have a look at strain. So there are Bragg edges that can serve in order to see either phase transformations or that we can use then to determine strains from a strain from by the Bragg peak position changes on the sample. Or you could, for instance, what is also, you can have a look at the microstructure in cultural heritage artifacts. That is also a field where these Neutron imaging methods are widely used. So our colleague Nikolai Kardylov has made a nice overview on that, a review article in Materials Today. So what are the recent tendencies on Neutron imaging? If you want, then you can have a look into that into more detail. Yes, thank you very much for your attention, and I would like to acknowledge all the people that have been involved in such a project. So there are people from ILL, of course, people from the UGA, people from the HZD, the former reactor in Berlin that contribute also to the new next instrument, as Sandro has shown. People from the ESRF here, the scintillator development, and people from helping with reconstruction codes from the KIT. Thank you very much, and I don't know if there are any questions. We can perhaps answer them, try to answer them.
Okay. Yes. Thank you, Lucas. Thank you, Alessandro, as well. Yes, we have still a bit of time for questions. I think that you could maybe raise your hands. It will be quicker than type in your questions, but you can use both. Waiting for some question. I have a question. Which is the largest sample that you could put on next?
We can we have a beam that is probably limited to something like a 200 times 200 millimeter square, so it's 20 times 20 centimeters. You can do field of view extension, for instance, in tomography, by placing the rotation axis close to the border of the field of view of the detector, and by this you can gain another factor of 1.7 or something like this. But in the order of 300 millimeter, I would say that is something that is a reasonable size, maximum size for samples.
That was in the old instrument. In the new instrument, we should be able to get to 30 by 30 by default. And also for tall objects like a blade, for example, a Viking saber, as we had before, you can essentially acquire a number of tomographies at different heights, and then you can compose them into long objects. It is possible to do relatively large objects in one axis. In the other axis, we are more 30, 40, let's say, in the new instrument. With this lateral stitching, we can do taking an image a bit to the right and a bit to the left of the object. Okay, thank you. Well, it seems that everything was clear. We don't have any questions. So I want to thank you, our speakers again. Thank you, Lucas and Alessandro. Thank you, Steph, for setting up the webinar. And thank you to all you to having come to this webinar. And please follow up Science in School, because there are some interesting one in the pipeline as well. Bye-bye to everybody. Have a nice afternoon and weekend.
Thank you.
Bye.
Thanks, everyone.